For the last year, I've been doing my master's thesis on the symbiotic interactions of fungi in native orchids. It's just been 1 year, so it's been pretty tough to fit everything in, but we've got some really good stuff. So I'll just introduce you to some New Zealand orchids. There are about 160 species of orchids in New Zealand, and only 9 of them are epiphytic. So there's this reliance on the mycorrhizal association. Many of our orchids are not showy, so we've got some very well camouflaged orchids, and on the far right there we have the Sporanthes, which looks very bright and colourful, but when we look at the whole plant, the colours kind of fade into the background, and it becomes cryptic in its environment as well. So almost a quarter of our orchids are at risk, with 10 of them being critically at risk. I've got 2 examples here. One is the terrestrial irwinii, which is known from 2 locations, and then this Anzibas or Corybas carciae, which is only known from 1 wetland location in New Zealand. So we need to do something about that. So the main threats are quite similar to the rest of the world. Habitat destruction is quite huge. Vegetation succession and competition, so invasive species and just natural processes, erosion. Herbivory, so we have quite a lot of mammal pests that are herbivorous, and then to a lesser extent collection, but that still occurs. It's important to understand these relationships so that we can relocate endangered populations or restore them to their natural habitat. And propagation is important for the conservation. We have the propagation program at King's Gardens, which is amazing to say the least, and it'll be great to emulate that kind of success in New Zealand. Bell, Newman, and Weihan taught me a lot of their methods, so there's kind of a dispersal of methodology happening as well. New Zealand is very behind in our orchid research. So this lady here, Dame Ella Campbell, she did a lot of work in the 60s and 70s with microheterotrophics, such as that gastrodia there. But she moved on to bryophytes, and since then we've had no real work on mycorrhiza on New Zealand orchids, and there's definitely no molecular work in the primary literature. So yeah, I went in and just found the field in New Zealand was wide open for research. So I decided to break it into three parts for my aims. Firstly was to describe the diversity of fungal endophytes, not just symbionts, in a range of New Zealand orchid species. And secondly was to identify which of these fungi do have the functional aspect and facilitate germination in vitro, and as a byproduct to find out which ones inhibit and consume the seeds or just inhibit germination, and then to take the in vitro experiments out into the field and see what we get in situ in terms of germination, and from a conservation point of view, trying to find viable methods to find viable sites to put these orchids once we do start propagation. So my methods were quite similar to what Nigel had, and he did a very good job of explaining that. Thank you. I chose these 6 species here for their contrasting distributions. So Microtis unifolia, similar to the Microtis media in Australia, is very weedy. It's everywhere. Pterostylis banksii and the Thalimitra, they're quite common in forests, coastal forests around Wellington. The Sympleglotis valida is actually a vagrant from Australia, and I decided to see if we could contrast those findings with the Sympleglotis cornuta, which is an endemic species, and see if there was any differences in diversity. I don't actually have my Sympleglotis results yet. It's still work in progress. Sorry, the Sympleglotis is a synonym for Chyloglottis, so there's a bit of taxonomy there. And then the Sporanthes nuovo-zelandiae is an endangered or at-risk orchid in a wetland in the lower North Island. The methodology was to isolate the pelotons, to rinse them, to culture all the fungi that I got, and then extract the DNA and sequence the ITS region with a range of primers just to ensure that I got all the diversity that I could out of this culturing method. There are obviously shortcomings where some fungi can't grow, or some fungi are competitively excluded by some more vigorous fungi at the start of the culturing process, or even consumed by them. So there's my graph. So from 191 sequences generated, I got 66 operational taxonomic units, so strains of fungi, you could say. And these were from Ascomycota, Zygomycota, and Basidiomycota. You can see the predominant orders that were represented in each of those phyla. And you can see that most of the diversity was from Ascomycota. It's just over half of that pie graph. I think it was 30 something. And secondly, it was the Zygomycota, and then the Basidiomycota are less represented. So from a functional point of view, traditionally the Basidiomycetes are the ones to look for. And I found one Ceratobasidium and 2 Rhizoctonia strains in the Terrestrialis, and one turned out to be Solani, which was the pathogen, which when I did my germination trials didn't work. And there were 3 Tillosnella strains isolated from the Sporanthus Nouveau-Zelandia, which this included the Tillosnella calispora, which is a very common symbiont. To have a look and see if I've discovered any new or native species that haven't been investigated, I just set up this threshold of 97% match between the GenBank accessions and my own sequences, and I found that there was a very small percent of perhaps novel species of fungi, but you can see one green at the 94%. There's a green Ceratobasidium which hasn't been described or hasn't been entered onto GenBank, and it may have been described before, but none of the molecular work has been done. So we need to look into that and see if there's a new potentially functional partner there. And so to kind of dig a bit further and to ask more questions and to see what we're looking at, I broke down the diversity by the species. So we can see that Terrestrialis banksii and the Thalamitra longifolia, they have higher diversity than the Sporanthus Nouveau-Zelandia and the Chyloglottis. But what surprised me was the lack of diversity in the Microtus unifolia. At the bottom, you've got the distribution of each of those species. So there's widespread and common on the left, and the vagrant, which is not very widespread, and the range-limited Sporanthus Nouveau-Zelandiae on the right. There are also other aspects to these that might come into play. So the time of collection, the Sporanthus Nouveau-Zelandiae and the Chyloglottis valida were both in full flower when I collected the roots, and all the other 3 were actually just starting to set seeds. So there may be some variation, and these are questions that I want to look into later on about the difference, the number of fungi that are in it at different stages in each plant. So the second part of my thesis was to identify which of those fungi that I did isolate were responsible for germination. So I chose a subset of the species in part 1 based on seed availability and sterilized those seeds and got viability scores for them. As you can see, the FDA staining on the far edge there. And then inoculated the seeds of each of those species with one of 36 strains that I had isolated in the part 1. And then used Microtus unifolia as a kind of positive control. So I tested all of my 36 strains against this Microtus unifolia. And I'm still counting these, so there's a lot of things I can't show yet because I'm not sure of what I've got yet. But I would like to talk to some people about that, and after the talk would be great. So we have a major milestone. We've got the first germination of a native a nationally vulnerable native New Zealand orchid happening. So this is part of this conservation drive that I really want to get to focus on. So yeah, we see this different stages. So we've got stage 3 there with the pelotons forming, and stage 4 with the leaf primordium. And as soon as the leaf primordium formed, I moved them into the light to continue developing, and they started to green up and look really good. So yeah, this is stage day 109 again. And at this point, they stopped developing, which I found quite interesting, because I expected them to just be feeding on food from the mycorrhiza, but they seem to stop. So my advice was to start to transplant them when their leaves got to the top of the plate, but it didn't actually reach there, so I was starting to panic a little bit. So I thought maybe the nutrients exchange might not be so important for the mycorrhiza. It might not be dependent on the mycorrhiza at this stage, so it would be time to plant them into the soil. So I got soil from a wetland and sterilized that, and just put the seedlings into the soil and a lot of water, and they seem to be working very well now. So there's some success, which is looking really good, even if there are only 20 or so plants, but it's a start. And then for the third part, we're looking at germination patterns in the field. So we use the seed baiting technique to identify or to have a look at whether or not the percent of germination varied depending on how far away from a parent plant you were. Packets of seed, one side contains Microtis unifolia seed, and the other side contains Thalimitra longifolia seed, between 2,000 and 3,000 seeds per packet, and buried them at various distances from established Microtis and established Thalimitra plants. There's a very, very low number of replicates. And yeah, so I'm still working on this, counting all the seeds, trying to get some statistical information out of that. We're getting what looks like very low germination rates, which I'd expect, but there's a lot of variability because of the low replicate number. One of my 4 plots in my negative control, I had a greater number of germinating seedlings, quite significantly. These 6 at the bottom that you see, all Microtis, they were all from one packet, whereas many packets didn't have any germination. Yeah, so I think they're all Microtis seedlings. I don't think Thalimitra worked in any of the plots. Yeah, and so essentially we have a high diversity in some orchid species, but we don't know why yet. We have to look into that. Most of the endophytes are from those 3 phyla, and there's no apparent link between endophyte diversity and the orchid abundance. But when I was looking at my sampling saturation, I would need to do probably a third more samples to get any kind of reliable, to get a very reliable number for the diversity counts. And then the success story is with the Tulisnella calispora, which promoted the germination of Nouveau Zelandier beyond stage 5. Yeah, and that just leaves me to thank Carlos Lenenbach, my supervisor, particularly Belinda Newman, and the people at Kings Park, and all those people for all the funds that that have been coming in. So thank you.